Okay, so on Friday we left off with Mendel's experiments, and so today we're going to pick up on the back side of the notes that we left off on, and let's see. My computer is slow. Okay, so Mendel's experiments, we talked about the dwarf trait he thought had been lost, right? And we talked about that was not exactly what happened. So biological inheritance is determined by factors that are passed from one generation to the next. Oh my goodness. Present. All right, sorry about that. Okay, so determine that factors are passed from one generation to the next. Today we know these factors to be genes. Okay, so each of the traits that Mendel observed in the pea plants was controlled by one gene that had occurred in two contrasting forms. For example, the gene for the height of the pea plant occurs in a tall form and in a dwarf form. And the different forms of the gene are called, who remembers, starts with an A, alleles. So Mendel realized that some alleles are dominant over other alleles. So this is where he came up with the principle of dominance and recessiveness. And that is not how you spell principle. It should be LE, of dominance and recessiveness. So some alleles are dominant and others are recessive. A dominant allele can cover up or mask a recessive allele. So dominant allele, if the dominant allele is present in an offspring, the trait will show up in the offspring. For a recessive allele, this trait will only show up if the offspring does not possess a dominant allele. So Mendel had another question. Had the dwarf trait or recessive allele disappeared, or was it still present in the F1 offspring? So Mendel allowed the hybrid tall offspring from the first generation to self-pollinate. So generation one of the offspring, tall, they self-pollinated. Okay, so we can see here's the parent, the original parent, right, was a tall and a short, and all of the offspring were tall, and that's what confused Mendel. So he was like, okay, I'm going to let those self-pollinate, plant the seeds, and this time he got 3 tall plants and 1 dwarf plant. He found that 3/4 of the offspring were tall and 1/4 of the offspring were dwarf. Evidently, the F1 tall offspring must have been carrying the dwarf trait, but it had been hidden. The dwarf trait had been passed down to the offspring, and it reappeared in the F2 generation, right, which is the second generation of offspring. Why did the recessive allele seem to disappear in the F1 generation, then reappear in the F2 generation? Well, Mendel realized that organisms have 2 alleles for every trait, right? You get one from your mom, one from your dad. These 2 alleles are inherited, one from each parent. If the offspring receives the dominant allele from one parent, that dominant trait will appear in the offspring. Recessive traits only show up in the offspring if the offspring receives recessive alleles from both parents or from each parent. Okay, so you have to, in order to show a recessive trait, you have to get a recessive allele from your mom and a recessive allele from your dad. In order to show a dominant trait, you only have to have one dominant trait from either your mom or your dad. You just need one. So if a parent has 2 alleles for a trait, how does the parent pass only one allele off to the offspring? Okay, this is what we talked about when we did meiosis. Today we know that answer to this lies in the type of cell division known as meiosis and the formation of gametes. Gametes are sex cells or egg and sperm cells. And you should recognize this picture, okay? So the letter T is going to represent our dominant allele, and our letter lowercase t is going to represent our recessive allele. So if the germ cell right here, which has both copies, okay, replication of DNA, now it has 2 copies. This is meiosis 1. It gets split into 2 cells. Then it undergoes meiosis 2, and those 2 get split into 4 individual sex cells. And each of those sex cells is haploid. So during meiosis, the DNA is replicated and then separated into 4 gametes. So we can see 2 of these gametes have dominant alleles and 2 of these gametes have recessive alleles. In this way, a parent passes one allele for each gene to their offspring. Mendel's principle of segregation. So Mendel's principle of segregation says that every individual carries 2 alleles for each trait. These 2 alleles separate or segregate during the formation of the egg and sperm cells. So homozygous or heterozygous. An offspring will inherit 2 alleles for a trait, 1 allele from each parent. The combination of alleles received by the offspring may be homozygous or heterozygous. Homozygous means that your 2 alleles are the same. Homo means same, so capital T, or little t. Heterozygous, hetero means different, so the 2 alleles are different. You have 1 tall t and 1 little t. So genotypes versus phenotypes. A genotype is the genetic makeup of an organism. A phenotype is the physical characteristic of an organism, what it actually looks like. So in this picture right here, we can see there's a homozygous tall plant and a heterozygous tall plant. Their phenotypes are the same because they're both tall, but the genotypes are different because one is homozygous dominant and the other one is heterozygous, meaning it has a big T and a little t. So, for example, in Mendel's pea plants, the tall allele was dominant over the dwarf allele. So he saw capital T gave you tall, capital T gave you tall, and little t gives you dwarf. So if we know the genetic makeup of parents, what type of offspring might they produce? So these are some famous families. What is the probability of producing different types of offspring? Okay, so here we can see different nationalities of people with different characteristics. So probability is what we call the likelihood that a particular event will happen. So now we're going to use probability and Punnet squares to work genetics problems. Okay, a Punnet square looks like this. It's very exciting. It's a box that's squared off into 4 little boxes. So a Punnet square is a diagram showing the allele combinations that might result from a genetic cross between 2 parents. The alleles of the first parent will be placed across the top of the square. The allele of this, just like that, okay? So big T, little t. The alleles of the second parent will be placed along the left side of the square. Big T, big T. The possible gene combinations of the offspring will be placed inside the squares.